Hey AP Bio subscribers. Today we're going to be talking about Campbell's 21.3. How exciting is that? So be sure to like, comment, and subscribe below. 21.3 talks about some of the other ways that um, other than natural selection, that a population can change. So, other ways a population can change besides natural selection. And there's really three major factors that can bring about evolutionary change. So, one of them we've already learned about is natural selection. Then we have genetic drift and gene flow. So, as a reminder, natural selection occurs when there is a differentiated success in reproduction, meaning some individuals are able to survive and reproduce more than others, and that in turn results in certain alleles being passed on to the next generation. The second one is genetic drift, and this change in population typically occurs when there's a very small population. Genetic drift is when alleles kind of quote unquote drift randomly and unpredictably from one generation to the next. And usually this results in the loss of certain alleles when we have small populations. So in this image, for example, if we take a look, if this was our whole population of flowers, it shows their alleles, it shows the frequency of those alleles, but only the highlighted plants survive and or happen to reproduce. Okay, this is only a small population of about 10 flowers. So of those five, now you can see we have a change in the alleles. Okay, now let's pretend that out of this group, only two of these have offspring. They both happen to be red, homozygous dominant, right? Now, when those plants reproduce, all we end up with is the homozygous dominant trait. We've eliminated these other traits from the gene pool, the CW allele. And so, if you only have a small population to begin with, sometimes genetic drift can cause only a few traits to arise and be continued to pass on, and then you end up with a completely changed gene pool, and it happens very quickly. Notice this only took 3 generations. The founder effect is a type of genetic drift that occurs when a few individuals become isolated from a larger population. So, in a sense, they found a new population. They're the founders. And typically, if they're breaking off and forming their own population, the alleles that are going to be available are the ones that they came into it with. So they typically tend to have smaller allele frequencies that are different than those in the larger parent population, and it's just due to whoever made up that population. We also have a type of genetic drift called the bottleneck effect, and this results from a drastic reduction in population size due to sudden environmental change. So maybe there's a big hurricane or the ice age or something like that. And then the resulting gene pool may not reflect the original gene pool because so many individuals may have been wiped out. And if the population continues to remain small, it may continue to be affected by these random changes and movement of alleles. It's kind of like if you had a bottle full of marbles, all different colors, but then you pour them out of the bottle and only a few get to come out, only a few survive. Here's our surviving population. You can see now we have mostly blue, one white. We've completely eliminated the yellow. And as you can imagine, now this small population would probably also be subject to genetic drift because now you have a really small population. One common influence of the bottleneck effect is human activity. So, for example, in Illinois in the early 90s, human activity caused there to be a severe loss of habitat for the population of prairie chickens. And the surviving prairie chickens had very low genetic variation, and only 50% of their eggs hatched and survived. So this, we think, led to a loss of genetic variation and increased the harmful alleles that were present. So you can see here, we started out, they had so much space, and then in the early 1820s, and then by 1993, human activity had all but eliminated only a little bit of their living space. And so here you can see they had a huge population size in the early 1930s, but by 1993, we had less than 50 prairie chickens left. This created far less alleles per gene locus and then resulted in very few of their eggs hatching, less than 50%. But where we had no bottleneck, these prairie chickens continued to thrive and survive and reproduce because they still maintained a large set of genetic variation. So genetic drift is very significant when the population is small. It can cause allele frequencies to change very rapidly and at random. It can lead to a loss of genetic variation within populations, and it can cause harmful alleles to become fixed and more permanent in the population, resulting in fewer surviving and reproducing individuals. Then we have gene flow, and gene flow is the random movement of alleles among populations. This can happen through the movement of fertile individuals. So if a fertile group of individuals moves to a new place, or maybe the gametes move to a new place, like pollen. And gene flow tends to reduce genetic variation among populations over time. So as time goes on, you have less variation. It can also decrease the fitness of a population. So, for example, there's a bird, the Parus major, on the Dutch island of Vlieland. And when birds immigrated from the mainland, they would bring alleles that decreased fitness in island populations, that made their fitness lower. And then natural selection acts on these less fit individuals and reduces the frequency of these alleles where immigration is low. But then when there's high immigration rates in the central population, the mainland immigration was overwhelming the effects of selection. So the gene flow increased these alleles even though they were deleterious. So you can see in this graph, the survival rates of the central versus the eastern birds. Let's see if this, let me look at this graph. You can see here, females born in the central population had far lower survival rates than the females born in the eastern population due to that gene flow. Another example of gene flow increasing the fitness of individuals could be seen in the spread of resistance to insecticides. So we began using certain insecticides to target mosquitoes that carry West Nile virus and other bad diseases. But of course, then we tend to see the evolution of alleles that evolve in populations which allow them to have resistance to these insecticides. And then these mosquitoes can survive and reproduce even when sprayed with the insecticide. And if they fly somewhere else, then they can increase the fitness of that population and begin surviving and reproducing there. And then eventually all the mosquitoes are resistant to that insecticide. So this is a very important agent of evolutionary change in modern human populations. So that ends at 21.3. Thanks for joining. You'll have these blue light glasses to help with all this screen time. Hope you like them. Like, comment, and subscribe below, and we'll see you